Every 3 seconds, somewhere in the world, someone develops dementia. Tens of millions of people are living with Alzheimer's, Parkinson's, or Huntington's disease right now. And the sad reality of all of these diseases is that medicine can slow them, but it cannot stop them. And it also cannot undo the damage they leave behind. A team of researchers in Japan, however, believes they have found a new angle of attack, and it starts with vitamin K. Hi, my name is Soumya Pillai. Welcome to another episode of Pure Science. To understand why this study matters, first we have to understand what these diseases actually do. Alzheimer's, Parkinson's, and Huntington's slowly damage the brain by destroying neurons, which are the cells that carry messages through the nervous system. Think of neurons as the wiring system in any unit. Every thought you have, every memory you hold, and every movement your body makes is electricity traveling along that system. When the wiring starts to burn out, the electricity being transmitted through it starts getting impacted. Thus, now in real terms, memory becomes unreliable, movement becomes difficult, and the very process of thinking slows down. As more neurons die, the situation keeps getting worse. What makes this so devastating is that the brain has a very limited ability to replace what it loses. Even in cases that it does, the recovery is nothing close to the rate at which these diseases destroy it. Current medicines can ease some symptoms, and recent Alzheimer's therapy can slow decline in certain patients with early disease, but they do not restore lost memories or rebuild damaged brain tissue. This is the gap that the researchers at Japan's Shibaura Institute of Technology decided to go after. Their approach was not to develop an entirely new molecule from scratch, but to look at something the body already uses and asks whether it could be made more powerful. The molecule they zeroed in on was vitamin K. In recent years, scientists have linked vitamin K to brain protection and neuronal differentiation, which is the process by which immature, uncommitted neural cells become fully functional neurons. The brain contains a reserve of these immature cells called neural progenitor cells or neural stem cells. Under the right chemical signals, they can develop into working neurons. Vitamin K, it turns out, is one of the signals that can push them to that direction. The problem is that naturally occurring form active in the body called MK4 doesn't push hard enough. Its effect is real but modest, not nearly potent enough to drive the kind of regeneration you'd need to make a dent in a neuroregenerative disease. So the team decided to build a better version. They synthesized 12 hybrid compounds by combining vitamin K with components related to vitamin A, specifically retinoic acid, an active metabolite of vitamin A already known to promote neuronal differentiation. And this is where things start getting interesting. Vitamin K and retinoic acid don't just do similar things through the same mechanism. They work through completely separate biological pathways. Vitamin K acts through a receptor called SXR, while retinoic acid acts through RAR. And when the team tested these hybrid molecules, they found they preserved the biological activity of both. In other words, researchers had engineered a single compound that could simultaneously turn two separate keys, activate two distinct set of cellular machinery that both push neural stem cells towards becoming neurons. One hybrid molecule in particular stood out from the rest. The team called it novel VK, and it showed roughly 3 times higher neuronal differentiation activity than the control, along with significantly stronger activity than natural vitamin K compounds. 3 times more powerful at turning stem cells into neurons than what your body naturally produces. While digging into how it worked, the team made a second unexpected discovery. Their analysis pointed to metabotropic glutamate receptors, specifically a subtype called mGlu1, as a key driver of vitamin K's ability to trigger neuronal differentiation. Now, it is important for reasons beyond just this study. mGlu1 is already linked to synaptic transmission, the communication process between neurons, and mice that lack this receptor show motor and synaptic problems that closely overlap with the kind of dysfunction seen in neurodegenerative diseases. Identifying mGlu1 as a target gives the entire field a clearer signpost for where to aim future drug development, regardless of whether it comes from vitamin K or something else entirely. Then came what is arguably the most important test of all. A compound that works beautifully in a laboratory dish means very little if it can't actually get into the brain. The blood brain barrier is the body's most formidable security checkpoint. It is a tightly controlled biological wall that blocks the vast majority of molecules from crossing from the bloodstream into the brain tissue. Many promising drug candidates have failed right here. Novel VK crossed the blood-brain barrier in mouse experiments, showed a stable pharmacokinetic profile, and produced higher concentrations of active MK4 in the brain than the control compound. It gets to the right place in the right form at meaningful concentrations. Now, these findings are based on cell studies and mouse experiments, not human trials, and no vitamin K derived drug has yet been shown to repair the brain of people living with Alzheimer's, Parkinson's, or the Huntington's disease. The road from a promising result in a mouse to an approved human therapy is, of course, long, expensive, and littered with compounds that looked extraordinary in early stages and failed in clinical trials. But the reason researchers and clinicians pay attention to work like this isn't about what it proves today. It's about the direction it points to. Almost every existing treatment for neurodegenerative disease operates on the same basic logic: slow the damage, manage the symptom, and buy time. A genuinely regenerative approach, if it could be proven safe and effective in humans, would aim at an entirely different challenge. Not slowing the loss of neurons, but replacing or restoring them. That would be a fundamental shift in how we think about treating these diseases, not just managing them. Scientists rebuilt vitamin K from the molecular level up, fused it with the active ingredient of vitamin A, and produced something 3 times more powerful at growing new brain cells. Something that can cross the brain's own defenses and get to work where it's needed. Of course, it's too early to laud its results, but it is definitely a promising progress worth discussing. That was all from me this week. I am Soumya Pillai and you were watching Pure Science on The Print.